Okay, ladies and gentlemen, good day all. Uh, in this video, we are going to look at the sequence alignment in bioinformatics. So, introduction to sequence alignment. Sequence alignment is a method used to identify regions of similarity between biological sequences. So, in sequence alignment is actually used to compare the regions of the of similarity between biological sequences. These biological sequences can be DNA, can be RNA, and can be proteins. So let's say you have two proteins from two organisms, and you want to see the identity of the regions of similarity to see if there are any regions between the two proteins, if there are similarity between them. For example, you can get the same gene from bacteria, you can get the same genes from human, and if you want to check and see if there are regions that are similar, so is this sequence alignment that is the method that you are going to apply in bioinformatics. And very importantly, why do we do the sequence alignment? It helps to infer functional, structural, or evolutionary relationships among sequences. So if you now take a sequence of protein from bacteria and then you now take it from human and you align them, if you see that their level of similarity is very high or their percentage similarity is very high, so that is to tell you that they are related in terms of their function. And also at the same time, so if you have a protein or DNA sequence that have high percentage of similarity, it tells you that they have related function. Also, it also gives you an idea of the related structure. So that is one of the very important things in carrying out sequence alignment. So based on the similarity between the regions, it would give you an idea whether the proteins have the related function or have related structure. So there are so many proteins that you can see them across different species, but with the sequence alignment, it will give you an idea whether proteins have similar functions or they have similar structure. And also at the same time, it gives you an idea about the evolutionary relationships among the organism. So that is why we use sequences from different organisms, and from there, based on the regions of similarity, it will give you an idea whether that organisms have any kind of evolutionary relationships. So then, in addition, ladies and gentlemen, the sequence alignment is fundamental in bioinformatics for analyzing DNA, RNA, and protein sequences. So these are usually the sequences that we use or that we analyze in sequence alignment, either DNA, RNA, or protein sequences. So this is how we do it. As you can see, ladies and gentlemen, this is a typical example of sequence alignment. So we have different types of sequence alignment, and one of the types of the sequence alignment, we have multiple sequence alignment. So as we can see, why is it called, why is it that it is multiple sequence alignment is because you have more than two sequences. You see you have HRAS, NRAS, and KRAS. All these are different sequences. So as we can see, there are three sequences and they are aligned. And as you can see, the number of the amino acid are 60. It started from 1 to 60. And from 1 to 10, it is actually fragmented. We have 1 to 10, then we have 10 to 10 to 20, 20 to 30, 30 to 40, and 40 to 50. And as we can see, with this sequence alignment, this is how to translate the language. As we can see, wherever you have these stars, it means that this is what we call conservation score. So the stars here, it means conservation score. So whenever you do a sequence alignment and you realize that there are these stars, it tells you that they are conserved sequences, meaning that they are identical. So that is where if you start from beginning to the end, you can see that all the sequences have stars. So this is to tells you that this is 100% similar. So they have 100% regions that are similar between the biological sequences. And then if you have two dots, when you carry it out or when you do any sequence alignment and you observe that there is two dots like this, and realize that there is two dots like this here. So this is to tell you that you have conservative mutation. And if you have one dot, it tells you that there's a semi-conservative mutation. And when you have a bracket like this, it means that you have non-conservative mutation. And then if you have dash like this or hyphen, it means that there are gap. This gap means that they are not conserved sequence. So this is how to translate. Whenever you do a sequence alignment, you'll see something like this. And in the next video, we are going to do practical demonstration on how to do the sequence alignment. So what are the types of the sequence alignment? We have majorly we have two types of sequence alignment. We have pairwise and multiple sequence alignment. So if for pairwise alignment, it's trying to compare two sequences to find the best matching regions. So in pairwise alignment, we compare two sequences. And then we have multiple sequence alignment, which aligns three or more sequences simultaneously to identify conserved regions. So these are the difference between the two. And as we can see, ladies and gentlemen, this is where we have a pairwise sequence alignment where two sequences are aligned. This is the first sequence and this is the second sequence. And this gap, it means that it is not a conserved region. And this one, it's supposed to be, if it is the normal one, you see stars like this, meaning that they are conserved region. And then for the multiple sequence alignment, you see, whenever you aligned more than two sequences, so it means that that type of alignment is called multiple sequence alignment. As we can see here, we have four different sequences, and this is what you call sequence alignment. Sorry, multiple sequence alignment. So then let's look at the pairwise sequence alignment method. So the common methods in pairwise sequence alignment, we have global alignment and we have local alignment. So global alignment example, Needleman, Wunsch, and then for the local alignment, we have Smith, Waterman. So these are the two common methods that we applied in pairwise global alignment. So in the global alignment or global sequence alignment, it aligns the entire sequence end to end, suitable for similar sequences. So in the case of the global alignment, you get two sequences and you align them end to end. And then for the local alignment, you find the best matching sequences, which is useful for identifying conserved motifs. So we use local alignment for identifying conserved motifs and it's not end to end. Let's see the demonstration here. If you can see clearly here from the local alignment, we just aligned the best matching region. And as you can see, it's not end to end. You see, this is your 5' and this is the 5' for the other sequence. So they are not end to end. So that is why it is called local alignment. In local alignment, you just check the best matching region and then you align. But for the global alignment, you sequence or you align the entire sequences, and that is how it is here. You see, this is end to end. So these are the differences between the local and global alignment. So then the next thing is the multiple sequence alignment or multiple alignment techniques. So we have so many methods or techniques or tools that are used in multiple sequence alignment. You can do multiple alignment using cluster W. You can do it using models database. You can use it using mapped. So these are widely used for multiple sequence alignment. And this method improves alignment accuracy by iteratively refining the alignment. So multiple sequence alignment helps identify conserved domains and evolutionary relationships. So this is how we use sequence alignment, sorry, multiple sequence alignment. So as we can see, the different software for multiple sequence alignment, we have cluster.blue, we have Mozart, we have mapped. So these are the widely used multiple sequence alignment tools. And then in addition, these methods, it helps in identifying a conserved domains and evolutionary relationships. And this is a typical examples of how to do multiple sequence alignment. So we have four sequences here. So as we can see, they are aligned. And typical examples here, I think from the previous slide, I show you that whenever you have a stars, like here, it is an indication that you have a conserved region. You see, that is why here, if you check from sequence 1 to 4, they are all SS. And also, we have LL across all. So that is why you see stars. I think this is the language. Look at it here, where I clearly demonstrate this. You see, whenever you have the stars with conserved sequence, that is, they are identical. And if you have two dot, that's semicolon, it means that you have conservative mutation. And if you have one, it means you have semi-conservative. And when you have hyphen, it tells you that there are gap, meaning that it is not aligned. So let's look at it from here. So you see in first one, there is a gap. There is a gap. And in the second one, you see there are two dots. It means that there is conservative mutation. And then look at this one here. This one, there is only one dot. It's an indication that there is a non-conservative mutation. So ladies and gentlemen, this is how to translate multiple sequence alignment. So whenever you have a sequence, whenever you conduct or whenever you do a sequence alignment and you have these symbols, so try to know their meaning and what they meant for. So what are the applications of sequence alignment? So sequence alignment is used in gene annotation, phylogenetics, and identifying functional motifs, and it aids in predicting protein structure and understanding evolutionary relationship. So sequence alignment is also essential in designing primers and developing gene therapies. So this is some of the applications or importance of sequence alignment. It's used in gene notation, in phylogenetics, and also identifying functional motifs. And it also helps in predicting the protein structure and also understanding evolutionary history. It's also very important in designing primers and developing gene therapies. And also apart from that, as you can see, you can use this sequence alignment in comparative genomics. In comparative genomics, you have gene from one organism and you have it from another organism, so you want to compare it. So that's comparative genomics, and it's also important in phylogenetic studies. It's also used in hierarchical function annotation. It's also very important in interaction network, structural or structural comparison modeling. So all these are different gene identification and validation are all applications of sequence alignment. So what are the major alignment databases? So first we have GenBank. A GenBanks provide a comprehensive collection of nucleotide sequence from multiple organisms. So if you want to get a sequence for nucleotide, that is the nucleotide sequence for multiple organisms, so you go to GenBank. And if you need the information about protein sequence and functional annotation, then we go to UniProt. And then we have Humble EB hosts a variety of databases, including ENSBEL, ENSEBLE, Interpro, and Reactome for the diverse biological data. So these are the examples of biological databases that you can get sequences. So then the next thing is in conclusion and future direction. So sequence alignment remains a cornerstone of bioinformatics with ongoing advancement in algorithms. Emerging databases and tools continue to improve the accuracy and efficiency of sequence analysis. So future development aim to integrate large-scale genomics data for comprehensive biological insights. So thank you very much, ladies and gentlemen. If this is the first time you come to my YouTube channel, don't forget to subscribe and join the YouTube for more videos. Thank you very much and see you next.